I'm not good actually.
What happened?
Cough and cold from last 15 days.
So did you not go for RT-PCR?
No,
How long you have been suffering from?
actually it's been 15 days.
Oh, it's 15 days. You must go for this test, but it is very reliable and giving the result on real time. You must go for it.
What is real time and what is reverse transcripted? Because I have heard both of them. So what is the
How
difference?
long she might
But I only know basic steps of PCR. Do you know about like history or discovery of
Yeah,
PCR?
I know. Actually, PCR is a very revolutionized technology in the field of as biologists. So we must know about its history. So it was invented by Polymerase in 1985. It was mainly because of the tech polymerase enzymes.
So basic steps in PCR are like denaturing, annealing, and extension. In denaturing, what happens the double stranded DNA strand will get into single stranded DNA at the temperature of 95 degrees Celsius. Because of high heat, the double stranded will come to single stranded. Yeah.
Nice. So now let me explain annealing. Annealing occurs when the primers bind at 3' end of the DNA, which are single stranded.
Okay.
And there are two types of primer, reverse and forward. So that will help in amplification from both the ends of
So
the
it will bind
single
into
stranded.
both the ends.
Both the ends of. And one more step is this primers. This is the forward and the reverse. This helps in addition of Taq polymerase, which is the main enzyme, which helps in polymerization.
Okay, which means it will give a space for it to bind.
Tag polymerase to bind.
Okay.
And the last step is extension. And the thing is, in this step, the tag polymerase will bind to the 3' end of the primer and also adds the DNT base. And this takes place in the at 72 degrees Celsius. And this is the whole cycle of PCR.
And I guess the step will again repeat,
Yeah, it
right?
repeats for 20 to
So there are different types of PCR like RT-PCR, multiplex PCR, nested PCR, and anchor PCR. We will see each in detail.
So I am telling about RT-qPCR, which is reverse transcriptase. Q signifies for real-time PCR. It also used in a COVID detection and other viral detection. So RT-qPCR done in a two ways. One is one-step RT-PCR and the other is two-step RT-qPCR. So in one-step RT-qPCR, at first mRNA is taken and it is being placed in a tube, in which we also add reverse transcripted enzyme which convert mRNA into complementary DNA. This process is called reverse transcription process. And while in the same tube, cDNA is being amplified and to form the DNA, which is also known as the qPCR, which is real-time PCR, and this will give in the software called thermocycler. So here we add all transcriptase DNA polymerase primer and mRNA. These all are being placed in a one tube only. While in the two-step RT-qPCR, we add RNA, mRNA, DNA polymerase, and the reverse transcriptase enzyme in one tube, which convert mRNA into complementary DNA. No, this process is also known as reverse transcription. Now this cDNA is being again placed in the other tube, which is then amplified. So now we have seen the one-step RT-qPCR and the two-step RT-qPCR, and now we know the difference. So where the advantage of the RT-qPCR, one-step RT-qPCR is that it needs the less pipetting and it contains the less contamination. While in the two-step RT-qPCR, there is a chance of contamination as it needs more pipetting steps.
Another type of PCR is multiplex PCR. As we know, in conventional PCR, in which we will add only one set of primers, which consists of reverse primer and forward primer. But in case of multiplex PCR, we can add more than one set of primers, like a maximum number of four set of primers. Total, it will be 8 primers. For example, how it works means, if we are given a chromosome and it has 3 genes, and when we add primers according to the sequence, and we can amplify the 3 genes at the same time. But in conventional PCR, what means we can only amplify one gene at a time. So that is the difference. And as we know, there will be disadvantage and advantage. Disadvantage is that while preparing the primers, all the primers temperature should be safe, because during the procedure like denaturation and annealing, the temperature should be safe. And another important point is that the primer shouldn't be complementary, which means if it's complementary, then those primers will be burned. So our process won't. Another type of PCR is.
Okay, I'm going to explain about nested PCR. Nested PCR is actually a modification of general PCR, and which is designed in such a way to ensure more specificity and sensitivity. In this, we usually use 2 sets of primers, and we perform 2 PCR reaction. As normal, these have the template and the master list. Master list composed of DNTPs, Taq polymerase, MgCl2, and buffer. And this green is one set of primer and the red one is another. In this green primers that usually amplify the regions which are slightly away from the designated region, and the red